Recording in progress.
Hello everyone. In this lecture, we are going to study about thymus and the T cell development. As you all know, along with the bone marrow, thymus is also a primary lymphoid organ because inside the thymus, the T cells will get matured. So if you see, this is the bone marrow. This is the bone marrow. And in the lecture of normal hematopoiesis, I have told you, all the hematopoietic cells will get originated from the pluripotent hematopoietic stem cell. So if you see here, this is the hematopoietic stem cell. Initially, it will give rise to the common myeloid progenitor and the common lymphoid progenitor. We're not going to discuss about the myeloid lineage here. If you see the lymphoid lineage, the common lymphoid progenitor, it will again differentiate into three lineages. One is the NK cell lineage, the second one is the B cell lineage, and the third one is the T cell lineage. Okay. So it can differentiate into the T cell lineage. And this differentiation into the T cell lineage happens because of a very important ligand that is known as Notch ligand. So this Notch ligand, this is very important for differentiating into the T lymphoid lineage. Okay, so basically these lymphocytes, they will have the Notch receptor, and when this Notch ligand will bind to this Notch receptor, they'll give signal to the lymphocytes to differentiate into the T lymphoid lineage. If Notch receptor or the Notch signaling is not present, they'll go for the B lymphoid or the NK lymphoid lineage. Okay, so that is why this Notch signaling is very important for the T cell lineage differentiation. Okay, so once this lymphocyte is programmed to develop as T lymphocytes, from the bone marrow, these immature T lymphocytes will go to the thymus. Okay, so if you see here, this is the thymus. This is the thymus. And these immature T lymphocytes from the bone marrow, they'll go to the thymus. Okay. Inside the thymus, these immature T lymphocytes, they'll mature into the mature T lymphocyte. So this is the mature T lymphocyte. Okay. And this maturation happens from the cortex to the medulla. Okay. So this maturation happens from the cortex to the medulla. We'll discuss in detail about this maturation process in some time. Once the T cells are matured inside the thymus, they'll come out of the thymus into the systemic circulation. Okay. So they'll come into the systemic circulation. In the systemic circulation, the T lymphocytes, they constitute around 60 to 70% of the lymphocytes. Okay. Again, this number is important. So in the circulation, T lymphocytes constitute 60 to 70% of the lymphocytes. B lymphocytes are around 20 to 30%, and NK cells are around 10%. Okay. And from this circulation, these T lymphocytes can go into the secondary lymphoid organs like the lymph nodes and the spleen. In the lymph node, they will populate the paracortex area. This we have already discussed. And inside the spleen, they will populate the periarteriolar lymphoid sheath. Periarteriolar lymphoid sheath. Okay, so this is how the T cells matured and go into the circulation and the secondary lymphoid organs. If you see the structure of the thymus, thymus is actually a bilobed organ. It's a bilobed organ. And you can appreciate that the left lobe is slightly larger and slightly higher than the right lobe. Okay, so if you see here, this is the left lobe and this is the right lobe. The left lobe is slightly larger and slightly higher compared to the right lobe. Okay, thymus is present in the anterior superior mediastinum, just behind the sternum and above the pericardium of the heart. Okay, and you can also appreciate that it also has a capsule. This also has a capsule, and this capsule is again, it is extending projections into the thymus parenchyma. These are known as the trabeculae, and it is separating the thymic cortex into various lobules. Okay, so this is same as we have discussed for lymph nodes and spleen also. We have discussed that lymph nodes and spleen will also have connective tissue capsule, and that will divide the lymph node and spleen into multiple lobules. Similarly, for in thymus also, this connective tissue capsule will divide the thymus cortex into multiple lobules. Thymus will also have cortex and medulla. So, okay, so this is the cortex area. This is the cortex area. and the inner part is the medulla. This is the medullary area. Okay. We have discussed, when we discussed about the lymph node, we have discussed that lymph node will also have cortex and medulla. But we have discussed that in spleen, there will be no cortex and medulla. Spleens are made up of white pulp and red pulp. But cortex and medullary, cortical and medullary differentiations are present in the lymph nodes and thymus. Okay. So these are some of the similarities and differences between these organs. One more point I want to tell about thymus is thymus will not have the lymphoid follicles. So no lymphoid follicles. And even if they are present, they are very minute in amount. Okay. Usually they do not contain lymphoid follicles. I mean the B lymphoid follicles. Lymphoid follicles are made up of B lymphocytes. Lymphoid follicles are found in the lymph node, in the cortical area. They are found in the spleen, in the white pulp area. However, lymphoid follicles are not present inside the thymus. Okay. The next thing is about the development of the thymus. The thymus develops from third pharyngeal pouch. Okay. So if you see here, these are the pharyngeal pouches. The first pharyngeal pouch will give rise to the auditory tube. This you must have studied in embryology. So first pharyngeal pouch will give rise to auditory tube. The second pharyngeal pouch will give rise to the palatine tonsil. It will give rise to the palatine tonsil. The third pharyngeal pouch will give rise to the thymus and the inferior parathyroid gland. And the fourth pharyngeal pouch will give rise to the superior parathyroid gland. Okay. So these are the structures that are derived from various pharyngeal pouches. You must have studied in the embryology. Okay. So thymus, it originates from the third pharyngeal pouch. Most of the times, this is the case. However, sometimes, very rarely, the fourth pharyngeal pouch can also contribute to the development of the thymus. Okay. Predominantly, it comes from the third pharyngeal pouch, but rarely it can also come. I mean, some component of thymus can also come from the fourth pharyngeal pouch. Okay. Significance of this development we'll discuss in some time. Okay. So basically, if you see the thymic structure, if you see the thymus structure, there are two cells that are predominantly present inside the thymus. The first one is the thymic epithelial cell. Okay. The first one is the thymic epithelial cells. Okay. These are the thymic epithelial cells. And the second one is the immature T lymphocytes. As I have shown here, these are the immature T lymphocytes. Okay. So basically, thymus will be predominantly composed of thymic epithelial cells and the immature T lymphocytes. Remember, these immature T lymphocytes are known as the thymocytes. Okay. So you should not be confused. Thymocytes doesn't mean the thymic epithelial cells. Thymocytes means the immature T lymphocytes that are present inside the thymus. And thymic epithelial cells are simply called as thymic epithelial cells, or sometimes these are also called as thymic epithelial reticular cells. Okay, so these are the two predominant cell types that you're going to find inside the thymus. Okay, so what are these two cells? First, I told it is the thymic epithelial cell, and the second one is the thymocyte. Thymocyte is nothing but the immature T lymphocyte inside the thymus. So thymic epithelial cells and thymocytes. These are the two predominant types of cells that you're going to find inside the thymus. Okay, and these thymic epithelial cells, these thymic epithelial cells, they originate from the pharyngeal pouches. Okay, so basically these thymic epithelial cells, they originate from the pharyngeal pouches. That means they have endodermal origin. Okay, so basically the thymic epithelial cells have endodermal origin. But the thymocytes, as we have discussed previously, the thymocytes, they come from the bone marrow, and bone marrow has mesenchymal origin. Okay, so bone marrow is of mesenchymal origin. So that is why thymocytes are of mesodermal origin. Okay, so thymocytes are of mesodermal origin. So from this discussion, you can understand that thymus has dual origin. It has endodermal component And the product of this endodermal component is the thymic epithelial cells. And it also has mesodermal component. And the product of mesodermal component is thymocyte or the immature T lymphocytes. Okay. So if someone asks you what is the development of the thymus, so you have to remember it is derived from both endoderm and mesoderm. The endodermal component will give rise to the thymic epithelial cells, and the mesodermal component will give rise to the thymocytes. Okay, and this endodermal component, it comes predominantly from the third pharyngeal pouch, but sometimes it can also come. It can also come from the fourth pharyngeal pouch. Okay. Though these are the two predominant type of cells that we're going to find inside the thymus, there are also some other cells which are found lesser in number inside the thymus, but we have to remember those cells also. So what are those cells? So basically inside the thymus, you can also find B lymphocytes. You can also find B lymphocytes in lesser number. However, these B lymphocytes, they will not make follicles. Okay, so they will not make follicles. Usually they present in the random fashion. You can also find dendritic cells. You can also find dendritic cells. You can also find the macrophages. You can also find the neutrophils. You can also find some neutrophils and some eosinophils also. Okay, so inside the thymus, you can also find the B lymphocytes, some of the dendritic cells, macrophages, neutrophils, eosinophils, and last but not the least, one of the very important cell type that you're going to find inside the thymus is myoid cells. So basically, these are known as the myoid cells. Myoid cells means muscle-like cells. These are muscle-like cells. Okay, and because of presence of this muscle-like cells, the thymus, because of presence of this myoid cells or muscle-like cells, the thymus plays a major role in the pathogenesis of myasthenia gravis. Okay, so because of presence of these myoid cells, thymus, or you can say the thymic hyperplasia, or sometimes the thymomas, they play a major role in the pathogenesis of myasthenia gravis. Okay, so myasthenia gravis is a neuromuscular junction disorder that we're going to discuss under the CNS pathology section. Okay, so these are the cells that you're going to find inside the thymus, but the predominant cells are the thymic epithelial cells and the thymocytes, which are otherwise known as the immature T lymphocytes. Okay. So as we understood this, now we'll go into little bit more details about the structure of the thymus. Okay, but before that, little bit about the anatomy. As I have told you, these are present in the anterior superior mediastinum. So these are present in the anterior superior mediastinum. Okay, just behind the sternum, anterior superior mediastinum, just behind the sternum and above the pericardium. Okay, above the pericardium. If you see the structure of the thymus, it will have a cortex. As I have told you, it will have a cortex. How can we identify the cortex? It will be, of course, it will be outer in location, and also it will be dark. Okay, so as you can appreciate here, these are it is dark. So cortex is dark. Why the cortex is dark? Because it will have abundant immature T lymphocytes. Okay, so cortex will have abundant immature T lymphocytes, and that will give a darker appearance to the cortex. However, the medulla is lighter in appearance because it will have lesser number of T lymphocytes. Okay, it will have lesser number of T lymphocytes. Okay, and inside the medulla, you will find a very interesting structure that is known as thymic corpuscle, otherwise known as the Hassel corpuscles. Okay, this we'll discuss in the next slide. Okay, so actually thymic corpuscles help us identifying this medulla. Okay, so they help us identifying the medulla of the thymus. So basically this thymus, if you see the weight of the thymus, what is the weight of the thymus? At birth, the weight is around 10 to 35 grams. 10 to 35 grams. It will gradually increase in size, and maximum size is obtained at around puberty, and the maximum weight will be around 20 to 50 grams. And this is achieved during puberty. Okay. And after that, it will gradually involute. And in the adults, we'll just find some remnants of the thymus, which will be around 10 grams. Okay. So the maximum size or the maximum weight of the thymus you can find around the pubertal age. Okay. And after that, the thymus is going to involute. And in adults, you'll just find some remnants and it will be around 10 grams. Okay. Sometimes the thymus can involute early in the development. So early involution of the thymus can be seen in some cases like severe illness, any severe illness or HIV infection. I mean, these are some of the conditions which can cause early involution of the thymus in the childhood itself. Okay. Then if you see, this is the X-ray. This is the chest X-ray of a, I mean, you can say it's a chest X-ray of a child or a newborn. In newborns, you can actually appreciate the thymus. These are present as triangular structures in the, in the mediastinum area. And this is known as thymic cell sign. Thymic cell sign. Okay. I mean, this is actually normal finding. This is a normal finding in the newborn. Sometimes you can appreciate this triangular shaped thymus inside in the chest X-ray. It can be present on the left side or on the right side. This is known as the thymic cell sign, which is a normal finding for newborns. In adults, you are not going to find this thymus. I mean, normally you are not going to find the thymus in the chest X-ray. Okay, so this is a little bit about the anatomy. Now we'll see into the histology or the structure of the thymus. So basically, as I have told you, it is divided into the cortex and the medulla. First, we'll discuss about the cortex. In the cortex, for the first point I have told you, it is dark. Why it is dark? Because it will have abundant thymocytes. So the thymocytes or the immature T lymphocytes, these are abundant. These are abundant in the cortex. So that is why they will give darker appearance to the cortex. Okay, so as you can see here, these are all the T lymphocytes. These are all the immature T lymphocytes that are present in the cortex. Apart from that, they will also have cortical thymic epithelial cells. Okay, CTEC means cortical thymic epithelial cells. Now, what are the characteristics of this cortical thymic epithelial cells? These cells, they will have abundant cytoplasm. Also, they will have cytoplasmic dendritic extensions. So as you can see here, these are the cytoplasmic dendritic extensions of the cortical thymic epithelial cells. Of course, they will have the nucleus, which I have forgotten to draw here. So they will have nucleus, of course, but they will have abundant cytoplasm. And these cytoplasmic dendritic extensions. Okay, and because of this cytoplasmic extensions, they can connect to each other. So two cortical thymic epithelial cells, they can interact, they can connect with each other through this cytoplasmic dendritic extension. And this cytoplasmic extensions, they will have tonofilaments. And with the help of this, they can interact with each other. Okay. Next thing is, what is the function of this cortical thymic epithelial cells? The first function. So if you see the function, the first function is they'll produce various factors, like they'll produce thymopoietin. They will also produce thymosin and thymic stromal factor. Okay, so the first function is they will produce various types of factors. These are known as thymopoietin, thymosin, thymic stromal factor, and there are various other factors also, which I am not writing here. So what are the function of these factors? These factors actually they will go to the circulation and they will attract the immature T lymphocytes from the bone marrow into the thymus. Okay, so basically these factors from the thymus they will go into the circulation, and in the circulation they will attract the immature T lymphocytes which are coming from the bone marrow towards the thymus. Okay, so basically they will attract the T lymphocytes into the thymus, and also they help in development of the T lymphocytes inside the thymus. So basically T lymphocytes development and T lymphocyte forming of the thymus is mediated by these factors. Okay. Apart from these factors, they will also produce the Notch ligand and interleukin 7. They will also produce Notch ligand and interleukin 7. These two factors, these two ligands, these two factors are also important for T cell development inside the thymus. Okay, so this is the first function. They will produce various factors that are required for the T cell development. The second important function of cortical thymic epithelial cell is they take part in the formation of blood thymus barrier. Okay, so what do you mean by blood thymus barrier? So basically, when in the cortex, this blood thymus barrier, it is found only in the cortex, not in the medulla, only in the cortex. Why this barrier is important? This barrier is important to prevent entering of circulating antigens into the thymic cortex. Okay, so basically this blood thymus barrier prevents unwanted antigenic penetration into the thymic cortex from the circulation. Okay, so if you see the structure of the blood thymus barrier, it is made up of, first of all, you will find the endothelium. So, okay, so these are the endothelial lining. So basically, you will find the endothelial cells with their basement membrane, of course. Okay, so endothelial cells with their basement membrane. This is the basement membrane. Surrounding this basement membrane, we will find the connective tissue. So surrounding that, we will find the connective tissue with some cells like pericytes and macrophages. Pericytes and the macrophages, which I have not shown in this diagram. And after that, on the thymic portion, you will find the cortical thymic epithelial cells and their extensions. So basically, this is the structure of blood thymus barrier. As you can see here, these are the endothelial linings. This is the basement membrane. This is the connective tissue, and also this connective tissue will be surrounded by various cells like the pericytes and the macrophages. And beyond that, we will find the cytoplasmic extensions of the cortical thymic epithelial cells. Okay, so basically, these cortical thymic epithelial cells, they are important in formation of the blood thymus barrier, which will prevent unwanted antigen penetration into the thymic cortex. Okay, and this blood thymus barrier is found only in the cortex. Okay, so these are the important points about the cortex. Now we'll go and understand about the medulla. So medulla, which is present in the inner part of the thymus, medulla will also contain thymocytes. Medulla will also contain thymocytes. The developing or immature T lymphocytes. However, these thymocytes that are present in the medulla, they are lesser in number. The number is less. That is why medulla is slightly lighter in comparison to the cortex. So cortex is dark and medulla is lighter. Why it is lighter? Because the number of thymocytes are less inside the medulla. We'll discuss in the next slide when we'll discuss about the T cell development. We'll discuss why the number of thymocytes are less inside the medulla. In the medulla also we'll find the epithelial cells. These are known as the medullary thymic epithelial cells. Okay, medullary thymic epithelial cells. The first point you have to remember is medullary thymic epithelial cells, they differ from the cortical thymic epithelial cells in their structure. Okay, so basically medullary thymic epithelial cells, they are spindle shaped. As you can see here, these are spindle shaped. These are spindle shaped, and they'll have lesser cytoplasm. Okay, so basically they'll have less cytoplasm compared to the cortical thymic epithelial cells. Okay, so they'll have lesser cytoplasm, and you can also appreciate that they do not have the cytoplasmic dendritic extensions. They do not have cytoplasmic dendritic extensions. So these are three important points of difference between the cortical thymic epithelial cells and the medullary thymic epithelial cells. Okay. So if I write here, the first point is these are spindle shaped. These are spindle shaped. They will have less cytoplasm. If they have less cytoplasm, they cannot have the cytoplasmic extensions. So they do not have the dendritic extensions. They do not have the dendritic extensions. Okay. At some areas inside the medulla, this medullary thymic epithelial cells, they'll make fold configuration. They'll make fold configuration inside the medulla. I mean, in some portions inside the medulla, this medullary thymic epithelial cells, they'll make concentric fold configuration, as you can see here. Okay, so this is the concentric fold configuration of medullary thymic epithelial cells, and these are known as Husserls. These are known as Husserls corpuscles. These are known as the Husserls corpuscles or thymic corpuscles. Okay, so these are nothing but the concentric fold configuration of the medullary thymic epithelial cells. Okay, and these are thought to be remnants of epithelial tubes of the pharyngeal pouches from which the thymus is developing. Okay, so basically these Husserls corpuscles are thought to be remnants of epithelial tubes of the pharyngeal pouches, especially the third pharyngeal pouch from where the thymus is developing. Okay, so this is the defining feature of a medulla. I mean the thymic medulla, where we are going to find these Husserls corpuscles. And actually, these Husserls corpuscles, these are keratinized. I mean the keratin proteins will be deposited in this Husserls corpuscles. So these are keratinized and easily identified on routine Hichani stain, where they will get stained as these will be stained like pinkish to reddish color. Okay, so Husserls corpuscles, these are keratinized structures. These are concentric fold configurations of medullary thymic epithelial cells. Okay, so this is about the medulla. So medulla will have lesser number of thymocytes. That is why they are lighter in appearance. They will have medullary thymic epithelial cells, which are spindle shaped. They will have lesser cytoplasm. They will not have dendritic projections. Okay, and sometimes, I mean, in some portions of the medulla, these epithelial cells, they will make concentric fold configuration. These are known as Husserls corpuscles. These are thought to be remnants of epithelial tubes of pharyngeal pouches, and these are keratinized. Okay, these are keratinized. So if you see the histology structure, if you see this this area first. Okay, so basically, this part is the cortex. The darker areas, you can appreciate these are the darker areas. This is the cortex, and of course, this lighter area is the medulla. So this is the cortex. This is the medulla. This is the medullary area. Okay, and inside the medulla, you can appreciate there are some pinkish to reddish corpuscles. These are known as the Husserls corpuscles. Okay, and in the hyper, you can appreciate that these are nothing but these are the concentric fold configurations of the medullary epithelial cells, and you can also find the keratin depositions in this concentric arrangement. Okay, so these are nothing but the Husserls corpuscles. Okay, so now that we have understood the structure of the thymus, now we'll understand the T cell development inside the thymus. So I have taken a portion of the thymus. So if you see here, just complete this diagram. So basically, this is a portion of the thymus. This is the cortex. This is the cortex area, and this is the medullary area. Okay, so this is the cortex. This is the medulla, and this area, the junction between the cortex and medulla, it is nothing but the corticomedullary junction. Okay, so this is the corticomedullary junction. So basically, this immature T lymphocytes or the thymocytes, they will enter the thymus from the circulation at this corticomedullary junction. Okay, so they will enter the thymus at this corticomedullary junction, and I have told you there are some factors. There are some factors which are produced by the cortical thymic epithelial cells, like the thymopoietin, like the thymic stromal factors, and these factors they will help attracting these immature T lymphocytes into the thymus. Okay. So the most immature T lymphocyte that is there inside the thymus, these are known as the double negative T lymphocytes. Double negative. Double negative means these are neither CD4 positive nor CD8 positive. Okay. So we have discussed previously when I discussed, I mean, in the lecture of hemophagocytic lymphohistiocytosis, I have discussed about the T cell activation. In that, I have told you predominantly T cells will have, I mean, these T cells are divided into either CD4 plus T cells or CD8 plus T cells. Okay. So either this, I mean, the mature T cells, either they will have CD4 or CD8 on their surface. Okay. But the immature T lymphocytes which come to the thymus, they will not have CD4 or CD8. That is why these are known as the double negative T cells. Okay. So after entering the thymus at the corticomedullary junction, these immature T cells first they will migrate to the outer cortex. Okay. So this is the outer area. This is the inner area. So from the inner part, from the corticomedullary junction, these immature T lymphocytes they migrate towards the outer cortex. That is the first step. They'll migrate towards the outer cortex. In the outer cortex, they'll proliferate. Okay. In the outer cortex, they'll proliferate. Okay. And after that, the actual development starts. Okay. So after this process, the actual development starts. So if you take out one immature T lymphocyte, as you can see here, this is one immature T lymphocyte. When the process of development starts, the first thing that happens is T cell receptor rearrangement. T cell receptor rearrangement. Okay. So T cell receptor rearrangement. What is this T cell receptor rearrangement? This I'll discuss in detail in the next slide. Okay. This I'll discuss in detail in the next slide. So basically, this T cell receptor rearrangement is required to produce different types of T cell receptors which can identify different types of antigen. Okay. So T cell receptor rearrangement is required to produce different types of T cell receptors which can identify different types of antigens. Okay. So the T cell receptor rearrangement will happen and that will produce, either the alpha beta T cells or the gamma delta T cells. Okay. So basically, the T cells based on the type of receptor that is present inside, or that is the receptor that is present over the T cells, they can be either divided into alpha beta T cells or gamma delta T cells, gamma delta T cells. Or there is one more rarer type of T lymphocyte that is known as NK T cell. That is known as NK T cells. Remember, the first thing that you need to understand is this differentiation of the T lymphocytes are based on the type of receptors that are present. Okay. So predominantly in the circulation, 95% of the T lymphocytes will have alpha beta T cell receptor. Those are known as the alpha beta T cells. Okay. So predominantly in the circulation, 95% of the T lymphocytes are they will have alpha beta T cell receptor. Okay. And those are known as the alpha beta T cells. 5% of the cases, they will have either gamma delta type of receptor or they can have NK type of receptor. NK type of receptors. Those are known as NK T cells. Okay. The point that you have to remember is these NK T cells are completely different from the natural killer cells, NK cells. Okay. So NK cells are completely different. The NK T cells will have some receptors. They will have some markers that are present on the NK cells. That is why these are known as the NK T cells. Okay. So inside the circulation, if you see the receptor, if you see the T cell receptor, 95% of the cases, 95% of the T lymphocytes will have alpha beta receptor. Okay. And the rest of the 5%, they will have either gamma delta receptor or they will have NK T cell receptor. Okay. So right now we are not going to discuss about the gamma delta and NKT because these are, they will be, they are actually very less in number. We are going to focus only on the alpha beta type of T cells. Okay. And these alpha beta T cells are divided into CD4 T cells or CD8 T cells based on the type of co-receptor that they have along with the alpha beta receptor. Okay. So 95% of the T lymphocytes in the circulation, they will have alpha beta receptors and they will also have one co-receptor. That co-receptor can be either CD4 or it can be CD8. If it is CD4 positive, that is known as CD4 positive T cells. If it is CD8 positive, that is known as CD8 T lymphocyte. Okay. Again, the exact structure of alpha beta receptor that we'll discuss in our subsequent slides. Okay. Now let us focus on the T cell development. So there will be T cell receptor rearrangement that will produce alpha beta types of T lymphocytes and also gamma delta type of T lymphocytes. This alpha beta T cell receptor. I mean, it is. Because of the T cell rearrangement, these T cells will have different types of alpha beta T cell receptors. Okay, they will have different type of alpha beta T cell receptors because these alpha beta T cell receptors, they are going to identify the. They are going to find the antigen. They are going to interact with the antigens which are presented by the MHC molecules. Okay, and because these T cells, they have to identify different types of antigens. These T cells, during this process of development, with the help of T cell receptor rearrangement, they are going to produce different types of alpha beta receptors. Okay. So once alpha beta receptors are formed, then they will produce the co-receptor. Then they'll produce the co-receptors. And these co-receptors, as I have told you previously, these are either CD4. These are co-receptor can be CD4 or CD8. But initially, the developing T lymphocyte will have both CD4 and CD8 co-receptor. Initially, the developing T lymphocyte, after it produces the alpha beta type of T cell receptor, it will have CD4 and CD8 molecule. And this is known as, this cell is known as double positive T cell. At this stage, this T lymphocyte, this developing T lymphocyte is known as double positive T cell because it has both CD4 and CD8. I have told you the most primitive immature T lymphocyte will not have either CD4 or CD8. That is why it is known as double negative. Now it has both. That is why it is known as double positive T cell. Once double positive T cells are formed, now the cortical thymic epithelial cell. So this is the cortical thymic epithelial cell. The cortical thymic epithelial cell will come and this will present the MHC molecules. This will, it will present the MHC molecules to this developing to this double positive T lymphocyte. Okay. So if this cortical thymic epithelial cell, if it can integrate the MHC class 1 molecule with the double positive T cell, the CD8 will be activated and the CD4 expression will be reduced. Okay. So for example, this is the MHC. This is MHC class 1 molecule. For example, this is MHC class 1 molecule. And we have discussed previously when I discussed about the T cell activation in the lecture of hemophagocytosis, lymphocytosis, I have told you the CD8 molecule can interact with the MHC class 1 molecule and CD4 molecules will interact with the MHC class 2 molecule. So basically this cortical thymic epithelial cell will express both MHC class 1 and MHC class 2 molecule. Some of the double positive T cells can integrate the MHC class 1 molecules with the help of CD8 co-receptor. Okay, with the help of CD8 co-receptor, some of the T cells can integrate with the MHC class 1 molecule. And those T lymphocytes, they will down-regulate their CD4 expression and they will ultimately, have only the CD8 molecule. Ultimately, they will have only CD8 molecule. Similarly, some of the double positive T cells can interact with the thymic epithelial cell with the help of MHC class 2 molecule. Okay, with the help of MHC class 2 molecule. Those T lymphocytes will maintain their CD4 molecule and they will down-regulate their CD8 molecule. Okay, so ultimately, two types of cells will be there. Ultimately, after this process, two types of cells will be there. One set of cells will have CD4 molecule and the other set of cell will have CD8 molecule. The other set of cell will have CD8 molecule. Okay, and this stage is known as the single positive stage. Okay, now these are known as the single positive cells because they have either CD4 or CD8, not both. Okay, so the cortical thymic epithelial cells, they will positively select the T lymphocytes to become single positive T cells. So basically, they will convert the double positive T cells into the single positive T lymphocytes. Okay, and this process is known as positive selection. This process is known as positive selection. Okay, so positive selection happens inside the cortex. So basically, there will be some double positive T lymphocytes which cannot interact with the cortical thymic epithelial cells. Okay, because of various reasons. This double positive T lymphocytes, some of the double positive, not some, most of the double positive T lymphocytes cannot interact with the cortical thymic epithelial cells, and those double positive T lymphocytes will undergo apoptosis inside the cortex itself. Okay, so this point is very important to understand. Out of the total immature, I mean, you can say out of the total developing T lymphocytes inside the cortex, around 90% of the developing T lymphocytes cannot engage their co-receptor, cannot engage their CD4, CD8 molecules with the MSC molecules presented by the cortical thymic epithelial cells. And those 90% of the T lymphocytes, they are going to die inside the cortex itself. They're going to die inside the cortex itself. So basically, this cortical thymic epithelial cells, they will positively select those double positive T cells, I mean, those developing T lymphocytes which can engage their CD molecules, I mean, either CD4 or CD8 molecules with the MHC molecules. Okay, so that is why this process is known as positive selection. And through this process, 90% of the developing T lymphocytes are going to die inside the cortex itself. That is why the T cells that are going to the medulla, they are lesser in number compared to the cortex. That is why cortex is dark and medulla is lighter. Okay, so I hope now you understood why the T lymphocytes, why the immature T lymphocytes are more inside the cortex and less inside the medulla, because 90% of them are going to die inside the cortex itself because they are not able to engage their CD4 or CD8 molecule with the cortical thymic epithelial cells. Okay, so this number is very important. Around 90% of the T lymphocytes are going to die here. Okay, and the positive selection happens inside the cortex of the thymus. Now, this single positive cell, they are going into the medulla. They will go into the medulla. Okay. Inside the medulla, the single positive T cells, they will interact with the medullary thymic epithelial cells. These are medullary thymic epithelial cells. So basically, medullary thymic epithelial cells are going to express, I mean, they are going to present various types of antigens of our body to the single positive T cells. Okay. Now you must be thinking how this medullary thymic epithelial cells is able to express, I mean, able to present various antigens present in our body. That is because of a gene that is known as AIRE, autoimmune regulatory gene. Autoimmune regulatory gene. We'll discuss more on this when we'll discuss about the immune tolerance, mechanism of immune tolerance, AIRE gene. With the help of this gene expression, the medullary thymic epithelial cells, I mean, they are able to produce, I mean, they are able to present antigens present in various parts of our body to this single positive T cells. Why it is important? I mean, why this process is important? Now, this is important because if this single positive T cell, I mean, if this mature T cell can interact with this self antigens, okay, if it can interact with these self antigens, those developing T cells has to undergo apoptosis. Okay, if they have strong interaction, if they have very strong interaction with these antigens, they have to undergo apoptosis. If they do not undergo apoptosis, when these T cells come to the circulation, they can interact, I mean, they can identify the self antigens and they are going to mount immune response against these self antigens that will cause autoimmunity. Okay, that will cause autoimmune reactions and autoimmune disorders. Okay, so if the developing T lymphocytes, if these single positive T lymphocytes, they are going to have a good, strong interaction with these antigens, which are produced by the medullary thymic epithelial cells, these T cells have to undergo apoptosis. So if they produce strong interaction, there will be apoptosis of these T lymphocytes. Okay, because these T lymphocytes are detrimental for us. If they come to the circulation, they will find self antigens as foreign. I mean, they can mount immune response against the self antigens. Okay, so this is how the things happen inside the medulla, and this is known as the process of negative selection. This is known as the process of negative selection. Okay, so the negative selection is the process by which the autoreactive T cells are damaged or destroyed inside the medulla itself, inside the thymus itself. Okay, so from this you can understand if there is a defect in this AIRE gene, there will be autoimmune disorders. There will be autoimmune disorders. Okay, so that I'm not going to discuss here. That we'll discuss when we discuss about the immune tolerance again under the section of immunology. Okay, so this name is important, AIRE gene. Okay, if they have only weak interaction, these T cells will survive. These T cells are going to survive because this weak interaction is not enough for T cell activation. So they are not going to harm our body. So definitely these T lymphocytes are going to survive. But if they have some amount of intermediate interaction, they'll have, they'll be converted to T regulatory cells. They'll converted to T regulatory cells. Okay, we'll discuss about the T regulatory cells. In a separate lecture, what are the functions of T regulatory cells and why these are important. But if they have intermediate interaction, most of the times they will be converted to T regulatory cells. If they have weak interaction, they'll become completely mature T lymphocytes. And if they have strong interaction, they are going to die inside the thymus itself. Okay, and these T regulatory cells and the mature T cells, they will go into the circulation through these venules with the help of a very important protein that is known as sphingosine 1 phosphate. Okay, not very important at this point of time for you to remember, but sphingosine 1 phosphate, this helps in exporting these mature T lymphocytes into the circulation. And I've told you inside the circulation, 95% of the T cells, they will have alpha beta receptors. 5% will have either gamma delta or NK T cell receptor. This alpha beta receptor, based on the presence of co-receptors, either they will be CD4 positive or CD8 positive, but not both. Okay, none of the mature T lymphocytes will have both CD4 and CD8. These T regulatory cells, most of the times, these T regulatory cells will have CD4 receptor on their surface. And along with this CD4, they will also have FOXP3. Okay, again, I'll not go into the details of T regulatory cells. Now, in this video, we'll discuss that separately. Okay, so this is how the T cells are developed or they're matured inside the thymus. Okay, and also this gamma delta T cells, they will go to the circulation. Gamma delta T cells will not have any CD4 or CD8 molecule. They do not need MHC. They do not need this CD4 or CD8 with MHC interaction to identify the antigens. Okay. So this is how things are happening. This is how the normal T cell development happens. Now I'll tell you a little bit more about the T cell rearrangement, how these things happens. So what is T cell rearrangement? First of all, what is the purpose of T cell rearrangement? T cell receptor rearrangement. What is the purpose of this T cell receptor rearrangement? As I have told you, this rearrangement is required because we want to produce different types of T cell receptor. Okay, so if you see, this is a T cell receptor. We want to produce different types and different shapes of T cell receptors. Okay, so these are all different types of T cell receptor. We want to produce different types of T cell receptor because we want to identify different types, thousands of types of antigens. So basically, T cells can identify thousands of types of antigens because they have diverse T cell receptors. Okay, so that this is known as lymphocyte diversity. This is known as lymphocyte diversity. This can happen only because of T cell receptor rearrangement phenomenon. Okay, this can happen only because of this rearrangement phenomenon. Okay, and these different types of T cell receptor, they can identify different types of antigens. Okay, now we'll see how exactly the things happen. So basically, if you see the T cell receptor gene, it has alpha gene. We are discussing about the alpha beta receptor, and we'll discuss about the rearrangement of alpha beta receptors. Okay, because that is the most common type of receptor you are going to find in the T cells. This T cell receptor gene will have alpha gene locus and beta gene locus. Again, inside this alpha gene locus, you will find various gene clusters, or you can say the DNA clusters. Okay, so if you see the alpha gene, this is the alpha gene locus. It will have various gene clusters. These are named as V, J, C. Okay, some of the DNA clusters are known as V DNA clusters. V means variable, so I'll just mention here. So V means variable, J means joining, C means constant, D means diversity. Okay, so these alpha gene clusters will have multiple segments. I mean, alpha gene locus will have multiple gene clusters or multiple DNA segments. Some of the DNA segments are variable DNA segments. I mean, they are named as variable DNA segments. Some of them are joining DNA segments. Some of them are constant DNA segments or constant gene clusters. For beta gene locus, we have B, J, C, and along with that, we also have D, that is diversity gene clusters. Okay, so these are the different gene clusters or the DNA segments that are there inside the T cell receptor locus. The important point to remember, these DNA segments or the DNA clusters, they cannot be transcribed independently as such. Okay, so these DNA segments independently they cannot be transcribed. That means these DNA segments, they cannot express, they cannot have their genetic expression independently. Okay, so how they can express the gene, how they can produce the T cell receptor, that is possible only because of this RAG gene. Okay, so basically, when the T cells, when the T lymphocytes, they will start to develop at this point of time here in the outer cortex, when they will try to, when they will try to mature, they will activate this RAG gene. Okay, I mean, this process also happens for B lymphocytes, not only for the T lymphocytes, it also happens for the B lymphocytes, where they have this B cell receptor realignment. Okay, the exact same process will happen there also. There also there will be activation of the RAG gene. So what will happen? This RAG gene, I mean, it has two components, so RAG1 and RAG2. So the point we have to remember here is this T cell receptor gene locus, the T cell receptor gene locus are present in all the cells of the body. I mean, all the nucleated cells of the body. However, this rearrangement and the ultimate genetic expression doesn't happen in all the cells. It happens only in the immature T lymphocytes. Okay, that is because this RAG gene is present. I mean, this RAG gene will be activated in these cells. What is the product of this gene? The product is VDJ recombinase. The product is VDJ recombinase. Okay, so RAG gene will produce VDJ recombinase. So this will cause, this recombinase has ability, to cause multiple DNA cuts and ligation in this alpha and beta gene locus, multiple DNA cuts and ligation. And this multiple DNA cuts and ligation, this will cause multiple combinations of DNA segment formation. Okay, so this will cause multiple DNA segment formation. Okay, they will produce multiple diverse DNA segments. Okay, and these DNA segments, they can be transcribed and ultimately they will form the T cell receptor. And because of this recombinase, there will be multiple CODs at multiple sites and ligation of multiple segments. So that will, of course, that will cause diverse type of DNA segment formation. That will ultimately cause diverse T cell receptor production. Okay. So this DNA segment can be transcribed and translated to produce the protein product, that is the T cell receptor. Of course, we are discussing about the alpha beta. So they will produce the alpha beta T cell receptor. This will also happen for gamma delta. It will also happen for the NKR T cell receptor, but we are only focusing on the alpha beta because that is what is more important. Okay, so this is the process of T cell rearrangement. This is the process by which they are able to produce different types of T cell receptors. Okay, so the last thing that in this lecture that I am going to tell you about is the alpha beta receptor. Okay, now we have discussed about the, we have we have talked about the alpha beta receptor. We have discussed about the co-receptors. Previously, we have discussed about how this, T cell receptors are interacting with the MHC molecules. Now we'll have a complete picture of the T cell receptor. So basically, alpha beta are the predominant component of the T cell receptor. However, this alpha beta, they present as a complex. They will present as a complex. That means along with alpha beta, you are going to find various other proteins that are there in the T cell receptor complex. So if you see the T cell receptor complex, okay, so you can see the predominant component or the antigen binding component is made up of alpha and beta subunits, which are joined by the disulfide bonds. Okay, so these are the disulfide bond you can see here. These are joined, this alpha beta chains are joined with each other with this disulfide bond. Of course, they will have a variable fragment. This fragment, this portion is variable. This portion is variable because they should be able to identify. They should be able to, they should be able to identify different types of antigens that will have. That is why they will have a variable segment, and on the lower side they will have the constant segment. That is the alpha beta component. But along with alpha beta, we will find various other proteins like CD3 proteins. So this epsilon gamma, this is one CD3 protein. This epsilon delta is another CD3 protein, CD3 proteins. So basically they will have multiple CD3 proteins, and also they will have a dimer of eta chains. So this is a dimer of eta chains. Okay. So this alpha beta heterodimer along with multiple CD3 proteins and eta chains, they will make the T cell receptor complex. I mean the alpha beta T cell receptor complex. Okay, so they will produce the alpha beta T cell receptor complex. Alpha beta along with the CD3 proteins and the eta chains. Okay, so this is the complete T cell receptor complex. Along with that, I have told you they will have either CD4 or the CD8 molecule. In this example, this is a CD8 positive T cell, or you can say a cytotoxic T lymphocyte. So it is having the co-receptor as CD8. Okay, so this is the CD8, and we have discussed previously CD8 molecule can only identify, I mean, cytotoxic T lymphocytes which are expressing the CD8 molecule as co-receptor, they can only identify those antigens which are presented with the help of MHC class 1 molecule. So this is the MHC class 1 molecule, and we have discussed previously MHC class 1 molecule will have unequal chains of alpha and beta. Alpha chain will be larger, which will have alpha 1, alpha 2, and alpha 3 subunits. And it will also have beta 2 chain, which is, I mean, it will also have beta chain, which is known as beta 2 microglobulin. The CD8 molecule can bind to the alpha 3 subunit of the alpha chain of the MSC class 1 molecule. Okay, so as you can see in this image. Okay, so these interactions we have discussed previously. In this diagram, I want to focus on the T cell receptor complex. So this is the entire T cell receptor complex. The complex is made up of alpha beta heterodimer along with CD3 proteins and eta chains. This will be joined by the co-receptor, that is either CD4 or CD8. Okay, and this interaction with the MSC class 1 and CD8 positive T cell receptor complex, this will give the signal 1, and there is another interaction between the presenting cell and the T cell, that is known as the B7 CD28 interaction. B7 otherwise the CD80 bar CD86. This interaction will provide the signal 2. Okay, again, I am not going to go into those details now, because this we have already discussed when I discussed about the T cell activation in the lecture of hemophagocytic lymphohistiocytosis. If you have not seen that lecture, you have to see that video to understand this signal 1 and signal 2 in more details. Okay, so one more thing I want to tell is all these videos that I'm publishing, all these videos are made sequentially. So you have to, it's better to see the previous videos to understand the current lecture properly. Okay, so this is about the T cell receptor complex, and this is about the alpha beta T cell receptor complex. That is the most common type of T cell receptor that we are going to find in the circulation. And as I have told you, 95% of the T lymphocytes in the circulation, they will have this alpha beta T cell receptor complex. And based on the co-receptor, they can be either CD4 positive T helper cell, or it can be CD8 positive cytotoxic T lymphocytes. Also, there will be a minor component of T regulatory cells. One more point I will tell here is the CD4 T cells, they constitute around 60% of the alpha beta T cells in the circulation, and CD8 T cells constitute around 30%. Okay, so number of CD4 T cells are higher than it is twice than the CD8 molecules. And this concept, I have told you the concept of T cell receptor rearrangement. This T cell receptor rearrangement will produce different types of variable region in the T cell receptor. Okay, so this variable region, as I have told you, variable region means that region of the T cell receptor which is binding to the antigen. Okay, so this T cell receptor rearrangement, the function of this rearrangement is to produce different types of T cell receptor Different types means they'll have different types of variable region. They will have different types of variable region, and through that they can bind to different types of antigen. Okay, little bit about the gamma delta and the NK T cells. Gamma delta T cells, these T cells, they do not need MSC. They do not need the help of MHC molecules to identify the antigens. So basically, they can identify peptide antigens, lipid antigens, and small molecules. They can identify peptide antigen, lipid antigen, and small molecules. Remember, alpha beta T cell receptor, it can identify only the peptide antigens. It can identify only the peptide antigens produced, expressed with the help of, presented with the help of MHC molecules. However, gamma delta T cells, they can identify peptide antigens, lipid antigen, and small molecules without the help of MHC presentation. These are predominantly present in the skin and mucosal epithelial cells of gut and genitourinary tract. Mucosal epithelial cells of gastrointestinal tract and genitourinary tract. Okay, so these are the sites for gamma delta T cells. Okay, so what are the points of difference between the alpha beta T cells and the gamma delta T cells? The gamma delta T cells can present, they can identify peptide antigens, lipid antigens, small molecules. Alpha beta T cell receptor can only identify peptide antigens. Gamma delta T cells are predominantly present in the skin, mucosal epithelial cells of GIT and genitourinary tract. But in circulation, predominantly it is alpha beta T cells. Rarely in circulation we find NK T cells. As I have told you, the first point is these are not same as NK cells. These NK T cells, they can identify the antigens which are presented with CD1 molecule. Alpha beta T cell receptor identify antigens presented with the help of MHC molecules. NK T cells identify antigens presented with the help of CD1 molecule. CD1 molecule is another MHC-like molecule. This is not exactly MHC molecule, but this is MHC-like molecule. This can present antigen to the NK T cells. And these NK T cells, they identify the glycolipid antigens. They will not identify the peptide antigens. Okay. So they will not identify the peptide antigens. They will identify the glycolipid antigens only if it is presented with the help of CD1 molecule. Okay. So in this lecture, we have discussed in detail about the structure of the thymus, the development of the thymus. We have discussed how the T cells are developed inside the thymus, and we have also discussed the concept of T cell receptor rearrangement, and we have also discussed the structure of T cell receptor complex. Okay, so all these points are very important with respect to immunology. We'll again revisit these concepts when we study the immunology.